Hi, my name is Esteban Miglieta. I'm a postdoctoral associate at the Simini Lab of the Imaging Platform at the Broad Institute. And today I'm going to talk to you about colocalization metrics. So this talk will be divided into two. On the first part, I'm going to briefly introduce what is colocalization, touch on some tips and guidelines for proper image acquisition, and then dive deep into different kinds of colocalization metrics. In part two, I'm going to demonstrate how to acquire these measurements, how to get all these metrics in different open source softwares such as ImageJ and Silverfiler. So let's begin with part one. If you ever worked with microscopy images at one point, you probably wanted to measure whether two different signals, two different labeled structures or molecules are co-localizing, that is present at the same coordinates as one another. So that's in very brief words why we define colocalization as a spatial co-distribution of two probes. In this example here, we can see that the vinculin stained in red and the actin filaments labeled in green are co-localizing. That is, like they are both happening at and these regions that we see yellow, that is the overlap of red and green, in this cell. However, so this is what colocalization is useful for. It's useful for determining the association of these two probes, that is the probe that labels vinculin and the probe that labels actin, in the similar region or space, that is the end of the actin filaments. However, this alone does not tell us whether these two molecules are interacting with one another. It can hint to it because, like, we know that in order to be interacting, they have to at least be close to one another. But colocalization and microscopy images in general alone are not enough to prove this. Why is that? Well, it's a limitation of the resolution of the technique. If we are using a regular confocal microscope, let's say with a 63x and 1.4 numerical aperture, we will be hitting a resolution limit of about 200 nanometers in the XY axis, of 600 nanometers in the Z axis. If we are using some more advanced super resolution technologies such as TED or Palm, Storm, we can get much lower than that, but still we are talking about the order of the tens of nanometers. Meanwhile, the actual size of GFP molecules, or of one generic fluorescent molecule, is in the order of just a few nanometers, so or at least an order of magnitude below the resolution limit. That means that when we are seeing one of these yellow pixels, that we are understanding, oh yeah, this means that these two molecules are in the same, occupying the same space. Actually, what we are seeing is a pixel or a voxel, if we are thinking it in 3D, that has dimensions that are much, much bigger than the size of the molecules that we are studying. So actually, what seems to be two molecules being in the same place can mean two molecules that are close together, like within this, for example, here 90 nanometer limit, but very far apart in terms of their own sizes. So this is why one has to be very careful when interpreting colocalization as saying, oh yeah, these two molecules are touching each other, are interacting with each other. It's a very good preliminary result to hint to something like that, but you will need further, probably biochemical assays to actually prove if such an interaction exists. Another characteristic of colocalization is that it's not a binary state. We can't just say, oh yeah, here these two probes are colocalizing and here they're not. Because if we see the channels here, it's very obvious. Most of the green puncta or the green puncta are corresponding to rect puncta. So we see all the overlapping image yellow. But here, for example, we see this cell is mostly green. So we would assume, oh yeah, there's no red fluorescent signal there, so it's not colocalizing. But when we see the red, we see that it is there. It's just that the relative levels of intensity in these two markers are different. So it is very hard, unless it's an extreme case just such as this, to categorically say, yeah, there is colocalization here. So we need quantitation. And for quantitation, we need appropriate metrics with which to represent this phenomenon of colocalization. And that's what we are going to be talking about. But apart from the quantitation, we also need some kind of statistical comparison. A number by itself tells us very little about the biology behind an image, because as we will see, many of these metrics, the raw numbers that many of these metrics produce are highly affected by technical circumstances or decisions that the analyst makes that have little to do with biology. So usually what you want to do is to establish some sort of comparison, either between a control and treatment, for example, cells treated with a drug versus cells treated with a vehicle, or some kind of biological specimen in time zero versus after some amount of time. On the other side, if you want to say, okay, but in a scale of not colocalized to perfectly colocalized, how is the colocalization in this one sample? You can always use compare it against a null hypothesis, for example, using the one sample student t-test. What I'm saying by this is there should always be some kind of statistical backup to the numbers that these metrics will produce. Furthermore, since we are going to be measuring microscopic images, there's a few things to take into account when acquiring these images so that they will be useful and they will yield like trustworthy results when we analyze the colocalization of the two probes. Since colocalization is a spatial phenomenon, so you want to see whether two fluorophores or two fluorescent signals are happening on the same space, you need to have good spatial resolution, as we mentioned before. So for that, it's highly recommended to use a high NA objective and also to set a proper pixel size and z-step if you are doing z-stacks using Nyquist criterion, which will allow you to achieve or to capture all the best resolution that the objective can give you. Another thing, since we are using and comparing two different fluorophores, you need to have good spectral resolution, that is to avoid any crosstalk or bleed through of the signal from one channel to another. How would that look like? For example, we have these two images. In green, we have labeled mitochondria, and in red, we have lysosomes. But actually, we see that there's some background in this red image, and if we crank the contrast up, we'll see that we can actually see the shape of the mitochondria, which can lead us to think that we are actually acquiring some of the fluorescence coming from the green label into the red channel. This should be avoided at all costs, because this will probably let you think that you are having a higher colocalization that you actually have, not because of the lysosomes, but because of these structures right here, which obviously colocalize perfectly with the green label. Moreover, again, because you want to have good spatial resolution and you want to see that you want to be only capturing the light that comes from the focal plane, you should use my imaging methodologies that avoid out of focus light, such as the convolution, if you are using wide field microscopy to get rid of that out of focus light, or confocal light sheet or turf or other imaging methodologies that only keep the in focus light. Another thing that you should check is that you have a correct registration of your image, and that is like correcting chromatic aberrations. Sometimes maybe because your objective doesn't have the correct corrections included, or maybe because of the setting or some misalignment in your microscope, something like this can happen where actually the green and the red channel are shifted one compared to the other, and that will again affect how you measure colocalization. Also, ideally you need bright and specific probes. That is, if you have a fluorescent signal, be it a fluorescent protein or like an antibody labeled, you need it to be bright with respect to the background. So you want a high signal to noise ratio, and you also need to avoid at all costs saturated pixels. You need to always work within the dynamic range of your microscope, and so if you have areas of saturation, those should be avoided when measuring colocalization, because you don't really know what is the real intensity of those pixels. So this is usually true for most measurements. When you want to make measurements in an image, avoid saturated pixels. And finally, it is possible to perform some sort of removal of unwanted signal or some pre-processing for making, for example, flat field illumination correction in your images prior to the measurement of colocalization. But it's very important that you then report what were the processing that were done to the image prior to the measurement. So how to quantify colocalization? Before going into how we measure colocalization, it is useful to differentiate two different phenomena that we are measuring when we assess colocalization, and those are co-occurrence and correlation. If you want to know a bit more about these, I highly recommend these two papers by Aaron and Dan, which delve much deeper into these concepts. So co-occurrence is defined as the extent of the spatial overlap between two fluorophores, whereas correlation is the degree to which the abundance of two spatially overlapping fluorophores are related to each other. This might be not very intuitive to understand at first, but we are going to go a bit deeper. But first, I want to clarify that whenever we are speaking about fluorophores, I'm actually prefer to speak about the signal that we detect from those fluorophores. So when we measure something in a microscopy image, we are not measuring the fluorescent molecules themselves, but rather the photons that we detected from those molecules that went through our microscope, our measuring tool. So it is not exactly the same. So that's why I'm usually going to be speaking about signal instead of the fluorophore. So try to put it a bit more simply. Co-occurrence tells us how much does signal A spatially overlap with signal B. So how much does it co-occur? How much of signal A is occurring in the same space as signal B? That's why co-occurrence metrics are usually best used to determine what proportion of a given molecule is present in a specific area or a specific structure, such an organelle or a cellular compartment. Whereas correlation tells us how much does signal A proportionally vary together with signal B. So how correlated are them to one another. So these types of metrics are most applicable when assessing a functional or possible functional stoichiometric relationship between these two species. So you're seeing how much they proportionally appear together. Whereas co-occurrence does not care about the proportionality of the signals, just as long as the signal is there. Let's go into the metrics to try to see if this is a little more clear. First, we're going to mention a metric of colocalization that is area overlap, also sometimes called area fraction, which is, as I put it here, is a measurement of co-occurrence alone. And we can see that because it is a quite simple metric. It ranges from 0 to 1. And basically, the procedure to obtain this is when you have two-channel image, you threshold each channel individually. And by threshold, I mean determine a pixel value to determine that all pixels under that specific value, that threshold, will be considered as background, and all pixels above it will be considered as foreground, or here displayed in white and black, respectively. And you do the same for the red channel, and you basically, from these two areas, you can get the intersection of the two signals and the union. Usually, whenever you measure the area fraction, you might get three different numbers, sometimes named K1, K2, and area fraction. K1 relates to the relationship between the intersection of both signals and in this case, the signal number 1 or green, or the intersection relative to signal number 2. And area fraction finally is the intersection over the union. You can see here that we don't care what is the intensity of the signals, just as long as they are above threshold. All we care is what areas are occupied by each other. This type of metric is, again, rather simple, goes from 0 when there is no overlap to 1 when there's a perfect overlap between the two signals, and is usually best used for really big objects that are well above the resolution limit of the microscope and are very well defined, such as, for example, nuclei. If you want to see if two nuclear signals happen in the same cells, well, this is rather easy to do. We can use another co-occurrence metric, which is object-based colocalization. And this is not so widely popular. It measures the position codependencies of the localization patterns. That is, basically, they take into account how near are the signals rather than necessarily overlapping. That's why this type of metric is very used for super resolution, and it's particularly useful for 3D data. Basically, it involves, again, setting a threshold for both channels. Then from those thresholded areas, get the centroid of each individual object or each individual area. So the point in space that is basically at the center of the object, and then setting some distance threshold to decide if these are closer together than random chance or not. Now we'll come to another metric, and it's probably one of the most widely used, if not the most widely used metric for colocalization, which is Pearson's correlation coefficient, or PCC. This is defined as the covariance of the red and green channel over the product of their standard deviations. I'm talking about here red and green, usually noted as R and G. This is just a convention, of course. It doesn't need necessarily be, it doesn't affect if it's red or green or any other color. Another way of expressing this same equation is that saying that PCC equals to the sum for each pixel in the image of the red intensity for that pixel minus the mean red intensity for the whole picture times the green intensity for that pixel minus the mean green intensity for the whole image divided by the product of the sum of squares of each pixel's intensity minus the mean. Basically, this means that this coefficient will be higher the more correlated the pixel intensities are. One easy way of seeing this, and this is something that usually accompanies the Pearson's correlation coefficient, is by plotting a fluorogram or scattergram. This is a plot in which each point represents one pixel in the image, and its X and Y values are its, in here, red and green intensities, respectively. So you can see from this, if the red and green intensities were perfectly correlated to one another, all these dots will be arranged in a diagonal line. So in that case, we would get a PCC value of 1, whereas in the extreme opposite case, if the red and green channel are anti-correlated, so whenever red is bright, green is dim, or vice versa, we'll get a value of -1. And in the middle, we get a value of 0 if there is no relationship, no correlation between the two channels. But there's some caveats to the PCC, and there's a very interesting, relatively recent paper discussing that actually the value we get and the interpretation of the PCC can vary a lot depending on where in the picture we are measuring it. So in this paper, they discussed that actually we can think of PCC as 3 different metrics. PCC all, in which the coefficient is measured for each and every pixel of the whole image. PCC or, in which we only measure we only take into account pixels that are either green or red, or here represented in magenta. And finally, PCC and, which is only measuring in the intersection of those pixels. So, for example, in this artificial image here, do you think, would you say that these two signals are colocalized? I would argue that yes and no, depending on where you are looking at. So here, for example, we see that if we use we use all the pixels, so take everything as foreground, don't exclude any pixel, we will get a PCC all of close to 0, 0.06. However, and that makes sense, right? Like most of the pixels in the image are black, so they don't contribute too much to this linear relationship. However, if we restrict the pixels to analyze and we say, no, we will only use the pixels that are either magenta or green or both, now we're getting analyzing only a very small subset of the image, only 17% of pixels, and we actually get a negative value of PCC for PCC or. And again, this makes sense because if you only pay attention to the pixels that are not black, you say, okay, yes, in the majority of pixels, so like 7 out of 8 blobs, they are anti-correlated, where you have green, you don't have magenta, and vice versa. But if you use PCC and, again, further restricting the number of pixels you are analyzing from the image to only 2% of them, you get a PCC of 1, a perfect correlation, because here you see all these blobs are brighter in the center and dimmer at the edges. And here, this perfectly white blob means that both magenta and green channels are bright at the center and dim at the edge. So they correlate perfectly with one another. If we go to a much a more biologically relevant example, we have this image in which we have two kind of vesicles labeled with green and magenta. Again, if we take all the pixels in the image, we get a PCC all of like close to 0. Again, understandably so, because most of the pixels are black. If we use PCC OR, we get a small but negative number. And if we use PCC AND, if you see those like a few white dots here and there, we get a positive, although again, low value. We can also see this in the scattergram, right? Basically, when we use PCC all, we are using like all the pixels in the graph for making this relationship. PCC OR excludes the pixels that are negative for both channels, and PCC AND only keeps this top right quadrant, that is pixels that are positive for both channels. So in summary, PCC all is measured on all pixels of the image. It depends on the co-occurrence and the proportionality of signals. So both, it's actually measuring both co-occurrence and correlation, but it's also affected by the ratio of foreground and background. And that has to do with non-biological phenomena, for example, like how densely plated were the cells in that particular experiment. Another option, PCC OR, it's measured on the union of all above threshold pixels. It also depends, although less, on the co-occurrence of pixels and correlation. So it is a mixture of correlation and co-occurrence, and it's more restrictive. It's measured on fewer pixels. Finally, PCC AND only depends on the proportionality of signals, because we are only measuring in co-occurring pixels. And it's the most restrictive, but it's also the most sensitive. So which one to use? Again, there's no correct answer. It's important that you know what you're using whenever you use it. I would say of all, the PCC all is the least recommended, because again, it's highly dependent on things that don't have to do with the underlying biology. But however, in most bibliography that I've ever read, it's the most widely used. Whenever someone uses Pearson's correlation coefficient, it is using all the pixels in the image. The PCC AND is the most sensitive to low levels of colocalization, so it's a good option if you have if you don't have like highly co-occurring labels, but you want to see, okay, in those places that they co-occur, how is the relationship to one another? And PCC OR, it's a mix of both. I would say is the simplest and most generally most recommended. But again, this is not a rule or anything. The most important thing here is to understand the difference between all these three metrics and report and like accurately report which one you were using and how. Another thing to consider when we are measuring PCC is which is our region of interest, or ROI. We can analyze any of the PCC and PCC all, PCC OR on the whole image as an ROI, or in different subsections of the image, such as here that we have 3 cells, and we can analyze the PCC, again, any of the PCC metrics independently on any cell like taking the whole cell as the unit. In this case, it's very interesting to see these two signals, red and green, are correlated to one another. You can see that wherever we have red signal, we have green signal, but there is the relationship, the stoichiometry of red to green is different. You can see that this particular cell is brighter than these other two in green, but not so much in red. What that gives us when we plot the fluorogram is that we have two different tendencies. Both are linearly correlated, so you actually have a high correlation in each cell. However, when you measure the PCC for the whole image, again, this can be PCC or even, or PCC and, you get a middling value of 0.6. Whereas if you were to measure in each cell individually, you and like get the mean of that, you would actually get a higher value, because PCC does not care about what the relationship is as long as it is a linear relationship. So that's something else to take into account when deciding how to measure your PCC. And also another thing that is very important to report so that your measurements are reproducible. Some other characteristics of PCC that we have to take into account is that Pearson's correlation coefficient is quite sensitive to signal-to-noise ratio. Here we can see in the higher, like having the same kind of correlation between the two channels, the more noise you get, the lower the PCC will be. And that is kind of expected, because noise is actual random signal. So if there is a relationship and you throw random signal on top of it, that relationship will get sort of messed up. And also, PCC is rather insensitive to background. This is true, however, only for PCC or and PCC and, as we have mentioned that PCC all can be affected by background. Another thing to consider is that areas with zero value pixels or saturated pixels, that is like values that are very extreme in the range, also can have an impact on this correlation. So this is the same thing I said before. We should always avoid saturated pixels. Zero value pixels will probably get red when we apply a threshold. Another characteristic that we sort of saw in the example before of the two cells with different relationships is that PCC assumes a single linear relationship. So any other kind of correlation that is not linear, so like if you plot the scattergram and you get any of these shapes, evidently there is no, these are not random, there is some structure to them, but it's not a linear structure. And so any of these will get you a PCC of 0. Of course, these are very rare in biology, but still. Here, for example, we have this red blob with a lot of green little blobs inside it, which you would guess should have a relatively high value for colocalization, because most of the green signal is within the red signal. However, that is actually just measuring the co-occurrence. The actual correlation, so whether the intensities of red and green vary together, it's all over the place, because here, again, red is brighter on the center and dimmer at the outside. However, all the green spheres are the same brightness wherever they are in the red. So you get a scattergram that looks like this, and if you only are measuring Pearson's correlation coefficient, you will see a very low value, which doesn't mean that you have low colocalization, just low correlation. So let's jump to another different metric from Pearson's, and that is Spearman's rank correlation coefficient. This is a metric that is very similar to Pearson's, but it actually addresses one of the weaknesses of Pearson's, and that is the one we just mentioned, that Pearson depends on a linear relationship. Spearman's can be thought as a non-parametric version of the PCC because it uses the ranks of the pixel values instead of the actual pixel values. So basically, if you have 1,000 pixels in this image, and you would put the highest green pixel, you would assign it a value of 1,000, the second highest a value of 999, and so on and so forth, completely getting rid of the actual values of intensities for those pixels. What this does is that whenever you have a linear relationship between the two channels, the SRCC, the Spearman's rank correlation coefficient, will give you a value that is practically the same as the PCC for the same situation. However, when you get a situation in where the correlation exists, but it's not linear, PCC will suffer a penalty for that, whereas the Spearman's rank correlation will continue seeing a correlation there and will still give a high value. This metric is also less sensitive to pixel saturation. So if you happen to have an image that is saturated, although ideally you shouldn't, this is better. And because of these strengths, some authors actually suggest that it should always be preferred to Pearson's correlation coefficient. Okay, now let's go to another widely used metric, that is Manders overlap coefficient, or MOC, or commonly referred to as Manders, although you'll hear in a bit that it's actually, we have to be careful when we say Manders to what we mean by that. So MOC is obtained by multiplying the intensity for each pixel in both channels divided by the product of the sum of the squares for each one. What that gives us is a metric that ranges from 0 to 1 and includes signal intensity distribution, so that like the co-distribution of both. So it's basically it's a mixture of co-occurrence and correlation. The opposite to what we said about Pearson's, this is quite robust against low signal to noise ratio. Here you can see in these situations with very with varying values for SNR. Here we have more noise. Actually, the MOC value varies very little. However, it is quite sensitive to background or offset. If you have this image right here, which has an MOC of 0.69, and we add on top of this some background that here it is exaggerated so for us to notice it, the image itself has not changed much, but however, the MOC is significantly higher. Another caveat with this metric is this is very biased towards the total area of the object in each channel. So for example, in this example right here, we have a very small green dot within a big red circle. And even though I would argue there is a high co-occurrence here because like all of the green signal is co-occurring with the red signal, the measured MOC for this is very low. Thus, some authors in the literature have done this Manders overlap coefficient, or MOC, as a confusing metric, and that is sometimes difficult to interpret, and so they don't recommend it very much. Manders also proposed other coefficients, which are called the Manders colocalization coefficients. And I say this in plural because it's actually two twin metrics, M1 and M2, which are kind of the two sides of the same coin. The MCCs are the sum of the colocalized pixels for each channel divided by the sum of all the pixels in each channel. So M1 is the sum of all the colocalized red intensities divided by all the red pixel intensities, and M2 is the same for the green. When I say colocalized pictures, I mean that this metric only takes into account the value of the red pixels in which the corresponding green is above 0. Actually, in general, what we will do is we will not use 0 as the threshold, but actually an actual threshold that we set. So basically, we will be computing all red pixels intensities in which the corresponding green value for the same pixel is above threshold. Let's see how this works with an example. So here we have these two images. In green and red, we see that they have different values for each pixel represented here in yellow within each one. And we want to see which pixels will be used for calculating M1 and M2. So first things first, we need to set a threshold. If you don't, we can just use 0. And in that case, all these 9 pixels within this square will be used for the calculation. If we set a threshold different than 0, let's say 11, in this case, I'm using 11 for both channels, but I could very well use different thresholds for each channel. So basically, when you have to calculate M1, you say, okay, this pixel, is it above threshold in green? No, okay, this I won't use it for calculating, or what is the same, I will assign it a value of 0 for the calculation. This number is the corresponding green above threshold. Yes, it is. Okay, so I add 14 here. And you do the same for all the pixels in the image. Another way then of describing what M1 and M2 are is that M1 is the proportion of red signal that co-occurs with green signal, whereas M2 is the proportion of green signal that co-occurs with red signal. And by signal here, we are referring to the accumulated above threshold pixel intensity. So the sum of all the pixels that are above threshold for that channel. So with these two metrics, it is easier to describe what was happening in this example where the MOC kind of failed. Here, we just have to say this object has a low M1 because the proportion of red signal that is co-occurring with green signal is minimum. Like most of the red signal is not happening in the same place where we have green signal. However, the M2 is quite high, and actually it is 1, that's the highest value it can get, because the proportion of green that is co-occurring with red is very high. All the green is co-occurring with red. So these two are informative of two different things. Depending on what you're interested, you can inform one or the other or both. Of course, M1 and M2 depend on which channel you're calling 1 and which channel you're calling 2. So that's something else you have to clarify. Some characteristics of the MCCs is that they are quite robust against noise, against low signal-to-noise ratios. Here you say with more noise, the value themselves vary very little. They do require you to choose a threshold, which is something that we've discussed before, but it will actually, it can very well change the value that you get, and is sensitive to background. So this is one very important take-home message, is that which threshold you select can have a really big impact in the value of the MCC and thus on its interpretation. There is Again, here, no correct answer on which threshold is the one to use, but it's critical that it is reproducible and thus that you can report it when you report your results. Because thresholding in colocalization analysis is so important, in their 2004 paper, Coste proposed a method for calculating the threshold automatically for images. And this method implies trying or testing decreasing thresholds for both channels until the value of PCC and, so for the whole image, of the pixels below the threshold is 0. So here, for example, you can see trying different threshold values and seeing what the PCC and is for the pixels below that threshold. And at one point, it dumps and reaches a value of nearly 0. So that's where this method draws the threshold. The logic behind this is that when you're calculating the Pearson correlation coefficient for pixels below the threshold, at higher threshold values, the pixels below will actually be part of your true signal, but the more you go down, you will get pixels that are mostly related to background, and where there is no biological correlation between the two channels, so that correlation becomes closer to 0, or it's a random relationship. We can also see this in a scattergram. Here, if we set the threshold value here for both channels, then we are using all these pixels to measure PCC, and of course we get a PCC that is higher than 0. The lower we get with threshold, we get PCCs lower and lower. So here it was 0.4, here it's 0.25, and when we get to this value of threshold, we get a PCC of 0, because there is like all these pixels are randomly correlated. So that's where you set the threshold. Is this perfect for all images? No, of course not. It actually performs rather poorly on images that are very poorly correlated, but it is reproducible. So it's good to try if doing a Coste automatic threshold is good for your particular image. Also, because it iterates through all possible threshold values, it can be computationally expensive, especially with 16-bit images. In their paper, Coste also proposed method for assessing the statistical significance of a given colocalization value. Take this image, for example. In both cases, we can calculate a co-occurrence of 0.9 between the red and green channels. However, it is easy to see that the value obtained here might not be necessarily due to an actual biological relationship between these two signals, but rather because there is a lot of red and of green signal in the image. And actually, what this method proposes is to scramble one of the channels related to the other and measure the same colocalization metric again and again and again for the scrambled channels. If you do that for these two images, you will see that all the colocalization values for the scrambled images are much lower than the actual value of 0.9 that we found before. Whereas in this case, the values for the scrambled images are very closely related to 0.9. So you can say that the value that we found for this is not statistically significant. It's not real. It's just due to random chance. So this is what we call Coste's randomization. Typically, in order to consider that the value of colocalization found is significant, it should be higher than 90% of the random iterations. And it can be applied to both PCC or an MCC or any other colocalization metric. And because it can be applied to anyone, it kind of should be because that way you can reinforce that the value that you found is real. One thing to take into account when doing the Coste randomization is that we don't just randomize each pixel individually. Here, for example, if we focus in, we see that it's not individual pixels, but blocks of pixels that are being randomized through the image. That has to do with that in microscopy images, due to the diffraction of the light, the value of one pixel is not independent of the value of its neighboring pixels. So it is better to set some sort of block size to randomize the pixels. Which block size is appropriate? That depends on the author. Some say it's the size of the PSF, the point spread function of the microscope you used for obtaining the image. Others say that it should be the diameter of the smallest object in the image. And others suggest that it should come from an autocorrelation analysis of the image. Again, whichever you choose, this should be mentioned or informed when you say how you obtained your results. Finally, I'm going to talk about a final metric of colocalization, which is not very widely used, but it is present in one of the tools that I'm going to demonstrate later, which is CellProfiler, and that is the rank weight colocalization coefficient. This is an algorithm that combines both co-occurrence and correlation, and basically what it does, it measures the correlation of all pixels, but it weighs that correlations on the difference of the ranks of the pixels in the different channels. So I talked about a lot of metrics. Again, the most widely used ones are Pearson's and Manders correlation coefficient. But also we have Spearman's rank correlation coefficient, which is similar and some argue better than Pearson's. So which one to use? Some authors provide some guidelines into which to use depending on different situations and which are the pros and cons of each one. But I would also suggest ¿por qué no los dos? Meaning measurements of correlation and measurements of co-occurrence bring complementary information. So depending on what is your biological question, maybe the best thing is to combine and to report more than just one metric. So some take-home messages from this very long talk so far. Colocalization is not a simple measurement. Most of the times it is not interpretable as a single isolated value. It should be compared to something else, either a control or do a statistical analysis to compare it to a null hypothesis. It does not imply physical interaction between molecules because, again, our images are limited to in the resolution that they have, and the acquisition is very, very important for getting reliable measurements of colocalization. Moreover, colocalization is a mixture of two different phenomena, that is co-occurrence and correlation, and measurements for both of these phenomena, such as Pearson's correlation coefficient, Spearman's rank correlation coefficient, Manders correlation coefficient, and any of the others that we mentioned, provide complementary information about colocalization and have different strengths and weaknesses that have to be taken into account when deciding which one to use. Also, the decision of which thresholding method or which threshold to use and the selection of ROIs have big impacts on the value of the metrics we measure, and thus on interpretation. So one of the most important things is it is very, very, very important to report what you're using and ideally why. Sometimes there is no why. Sometimes there is it's just an arbitrary decision to use this threshold instead of this other. But the most important thing is to report accurately report which one you're using in order for your analysis to be reproducible. And also, ideally, whenever possible, to perform Coste's randomization to robustize your measurements. With this is the bibliography that I used for this talk. This is very interesting if you want to dig deeper into some of these topics. Thank you very much. I'll see you in the next video.